The rest of the 70 to 95% water-containing cells consists mostly of carbon-based compounds. So what is so special about carbons? First of all, they're tetravalents, meaning that they can form 4 covalent bonds. The fact that carbon has 4 electrons in its outer energy shell allows it to form large chains and complex molecules with itself and other atoms. Having 4 electrons in its outer energy shell means that carbon wants to and can form 4 bonds. This enables carbon to form chains, branch chains, and rings. These structures wouldn't be possible if carbon could only form 3, 2, or single bonds. Also, there are certain shapes the bonds are formed: tetrahedral, bent, or linear. So without carbons, many large chains and complex molecules that we know of today, such as proteins, DNA, carbohydrates, would not exist. It's like we're chasing all those stars. First of all, let's talk about how cells synthesize through dehydration synthesis. Dehydration synthesis is the removal of water, the removal of the hydrogen molecule, in other words. This allows the remaining reactants to join together, thus being a synthesis. Now then, how do cells break down products? Cells break down products through hydrolysis. Hydrolysis is a chemical process in which a certain molecule is split into 2 parts by the addition of a molecule of water. While one fragment gains a hydrogen ion, the other gets the hydroxyl group. This process, while being the opposite of dehydration synthesis, breaks down polymers as well. Polymers are groups of combined monomers. Thus, breaking down polymers is also breaking down products. Therefore, dehydration synthesis takes place when the monomers of organic compounds join together by a chemical reaction to make polymers, while hydrolysis occurs when a polymer needs to be broken down. These are the ways how cells synthesize and break down products. Got no reason, got no shame, got no... Carbohydrates include both sugars and polymers of sugars. Carbohydrates serve as fuels. They're structured in a way so that they have a lot of carbons, and in other words, they have the most number of carbons in their structure compared to many others. This kind of structure is very advantageous for their function. Next, for lipids, I'd like to talk about 2 groups: triacylglycerol and phospholipids. First, the triacylglycerols. Their main function, to store energy, is helped significantly by their structure. Because they have 3 fatty acids linked to 1 glycerol molecule, again stressing the fact that they have 3 fatty acids linked, they complete their function well. Next, phospholipids are mainly function in the plasma membrane. They function well because of their structure as well. Phospholipids, having hydrophilic head and hydrophobic tail structure, is able to work well in their function. Proteins have a lot of function, and this is the result of their various structures. They act as storage areas, speed up reactions, and do many more. Enzymes, for instance, are mostly protein, and their structure allows them to work effectively in lowering down the activation energy. If their specific structures were changed, their functions would have been changed as well. Nucleic acids. Their structures of double helix, and with the members of the pyrimidine family and purines, cytosine, thymine, uracil, adenine, and guanine, they all account well with their function mainly related to hereditary information.